Yeah, so I'm Mark Perkins. I work for Analytical. I'm applications chemist, predominantly working on SIFT-MS. And so I gave 2 talks here, and the second one was more looking at the validation of these methods. So can we justify the results and see that they're robust and usable results? Well, the second talk was, building on the first one, which is, okay, we show that we can do this automation, we can generate these interesting analyses, we can use it as a more routine analytical device. But if you don't have confidence that the results you're generating stand up to scrutiny, then what use is that technique? So what I show with that second talk is that we can apply standard validation methodology, use the standard protocols that exist already and apply them to SIFT-MS methods, and show that the data we generate is as robust as any standard technique that is used by people out there. So really putting SIFT-MS on a sort of foundational footing with the rest of analytical techniques that lab users would use, really. By adding automation, by validating these methods, we take an instrument that people do look as being specialised, and what we're saying is, that it isn't a specialised instrument in that sense. I mean, really, that's what I feel my job is to take that instrument and say, it is an analytical device. We can use it as you would use any other instrument in the lab, but it has certain characteristics that makes it unique, not specialised, but unique in the way it does the analysis. And so don't be scared of it. It's just an analytical instrument. A very good one, but just an analytical instrument.
So our first speaker today, Mark, he's back.
I'm back.
And this time he's method validating SIFT-MS.
Yeah. So this sort of follows on a bit, I suppose, from yesterday's talk about automation, but it's about creating methods that are validatable on SIFT-MS, because you might, I have been asked a number of times, is it possible to validate SIFT-MS methods? It's quite an unusual technique compared to traditional sort of analysis techniques. So can we validate SIFT-MS methods? So we're going to go through this presentation is 2 sort of case studies. One which would be classed as a bit of more traditional SIFT-MS type method, which is gas phase analysis and what sort of... That's all right. So manual introduction of gas phase samples to the system. And the second one is more following on from yesterday's work on the stuff we can do with automation and a more sort of involved method prep method. So the first thing to consider with method validation on SIFT-MS is, to some extent, you've got to do a method development before you can validate a method. So very broadly and quickly, just what is the difference between sort of SIFT-MS method development and GC-MS method development? Well, the first thing to say really is that on a GC method, we get more choices than we get with SIFT-MS in terms of column, in terms of how you introduce a sample, how you program the oven for the temperature ramps, what detector to use. With SIFT-MS, we don't have a column, obviously. It doesn't separate by column, it separates by mass spec, and the detector is pretty much given to you. So our choices for method development of SIFT-MS is much, much simpler, in the sense it's just how we get the sample in, some of the automation stuff yesterday, and pretty much what reagent ions and product ion pairs we use to do the development. So the other thing to say, of course, about method development is it's much more quick on the SIFT-MS, because if you make a mistake in your method development, you've got your sample in the column at this point. It's going to take you maybe 20 minutes to run it back out the back end of the column before you can put the next one in. And so your method development process is somewhat slower with GC-MS. With the SIFT-MS, if you get a sample into your system and you've done it wrong, you've got the wrong concentration, you've made it the wrong way, pretty much you can stop the method there and then. The whole thing clears through the system. Any carryover, it's gone within less than a minute, and you can start again. So you can see how the method development cycle is much quicker with SIFT-MS versus GC-MS, which means that hopefully that translates into much quicker method validation as well. So first part of the talk is going to be on some work that wasn't done by us. It was done by Joe Wicks at Intertech Pharmaceutical Services, based up in Manchester. It was for a customer of theirs looking at a pharmaceutical application, and it was basically looking for formaldehyde in headspaces of a medical device. And this was done because the company needed to submit for a regulatory submission their formaldehyde concentration. So this whole method validation was done for a regulatory submission to the MHRA. So it had to work and had to work properly. So this part of the presentation was put together by Joe, and I'm pretty much presenting it as he presented it. So bear with me. So we can sort of move through the first bits. I didn't want to take them out as it is his presentation, but this is stuff we know about SIFT-MS, the analytes we can use, how it can look for some particularly difficult compounds. For example, formaldehyde in this case for this validation, and also applications, residual solvents in drugs, packaging, selection, compatibility, contamination in products, which is what this validation was about. And the reason they chose SIFT-MS was pretty much because it was highly sensitive to formaldehyde, and the formaldehyde was in the gas phase, and it made the measurement really quickly. So the process they used basically was to take the product, stick it on the SIFT-MS for 30 seconds, like manually stick it on the SIFT for 30 seconds, and measure the headspace concentration inside a closed pouch system. And that was their measurement. So it was a manual but quick process for formaldehyde. So the validation was about putting together the package of data to show that this method was robust and gave results that could be trusted. So we'll move on from this slide here, which is just some numbers about how you do gas phase analysis, which hopefully if you all do it, you should know how to do it. And we'll move on to the validation. So the question here is, how do we validate SIFT-MS methods? Well, for GC and LC, there are guidelines out there already, particularly for sort of pharmaceutical type validations that you can work to, which are basically the ICH Q2 guidelines, so the International Committee for Harmonisation of Methods or something, provide the framework for doing method validation. So it covers all the things you need to do, such as the linearity and the precision and the robustness. And so this method was validated using and following the ICH guidelines and making changes where necessary, because these are predominantly written for chromatographic type methods. And so this was, could we force or push a SIFT-MS method through this type of validation process? And so what we'll see over the next few slides is how Joe Wicks treated the SIFT-MS method and treated it with regard to this, these guidelines. So the first part of the ICH guidelines is specificity. So can you see your analyte in the presence of the matrix that sits behind the analytes you're interested in? So to sort of quickly just run through what ICH says, it says investigation of specificity should be conducted during the validation process to demonstrate that when we want to look for those analytes, we see them. So normally with a sort of GC type method, you would spike in your analytes into the matrix, run the method, see if there's any co-eluting peaks or anything that gets in the way, or do your peaks move about a bit. But with SIFT-MS, we don't have that. Everything comes out at the same time. So what we're trying to look for is when we see a product ion that says it's the analyte, is it the real analyte? And so the way Joe approached this was a formaldehyde method. He basically searched the library to see if there were any conflicting ions in the library that will conflict with formaldehyde. So, he searched the library, you can't see that very clearly, look for formaldehyde, look for the product ions that would look like formaldehyde. Is there anything else in the library that looks like that? So he based the specificity on no conflicts in the library. Now, clearly it worked because this method validation passed, and that they accepted that approach to specificity. I'm not so sure that's actually the only thing you need to do, because there could be things that aren't in the library that are in your matrix. And so I think actually what you need to do for specificity is a bit more involved. You need to run some of your matrix, some of your standards. You need to run it in full mass mode using the various reagent ions at your disposal, and just to see if there are any conflicts that sit behind you hadn't expected to see. Run your blanks without the analyte, and then really dig into what is happening in the matrix, because the one thing we don't get with SIFT-MS is we don't get separation in time. So if you've got conflicting stuff in there, you have to work hard at finding it. You can't just pull it apart on a chromatogram and say, look, there's the peak. So there's more involved in specificity, but in this instance, I think with formaldehyde, because it's a low mass analyte, there isn't a lot actually to get in the way at that mass it's being detected at. So in this instance, it works. But for higher mass VOCs, I think you'd have to work harder to get there, to be honest with you. But they accepted it, so we're not complaining. So the next part of ICH guidelines for method validation, linearity, obviously. So linear relationship should be evaluated across the range of the analytical procedure. So how did they do this in this case? So this was a gas phase measurement. They were measuring formaldehyde in pouches. So basically they produced their formaldehyde gas standards by using a permeation oven. So with a paraformaldehyde in an oven, passing gas across across the permeation tube and running that gas into the SIFT-MS. And by changing the flow rate through the permeation oven, you can change the concentrations of the formaldehyde. So they generated linearity curves, basically, and range curves by using the full range of the permeation oven. And this was the first set of data they ran. So they ran it from about 100 ppBV up to 35 ppm. The expected concentration in their devices when they were looking to analyse was somewhere between 2 and 5 ppmV formaldehyde. So this clearly covers the range of expected concentrations. The other thing you can see on this table is that we've got the measured concentration versus the theoretical concentration, and then here the concentration as calculated off of that calibration curve. So if we use that calibration curve to back calculate it, what will we get? And this generates these recoveries here. And you can see at the low concentrations, we're massively over-recovering according to our calibration curve on this linearity. And the reason for this, if you look closely, is this point up here, the 35 ppmV point, is actually pushing off the curve a bit. And because it's such high concentration, it actually carries a significant amount of weight in that linearity, and so it actually skews the bottom end of that linearity curve. And this is where you're getting this over recovery, because the line is not actually running true through those bottom points of the linearity. So 35 ppmV didn't need to go that high for the method anyway, and it probably was pushing the top end of the SIFT sensitivities at that point anyway, or the SIFT limits. So they reran the linearity, took out that top point, ran it up to 15 ppmV basically, and you can see now that all the recoveries based on that linear curve are now somewhere below plus or minus 20% of the recovery you would expect to see. Clearly a nice linear relationship. So the system is linear across, what's that? That's 3 or 4 orders of magnitude for formaldehyde, with a R squared of 9998, which is really good for a difficult analyte. So we've proved linearity. So the next thing to do, precision. So precision of the instrument. How do we do that? Well, basically you generate some standards and you run them and you see how precise that is. So ICH says basically repeatability should be assessed as a minimum of 9 determinations covering the specified range of the procedure. So 3 replicates of 3 concentrations. So in this instance they did 6 measurements, 3 preparations at 50% of the linear range. So they used their gas standard generator, set it to give a fixed concentration of formaldehyde, filled up some Tedlar bags with the gas standard, put the bag on the instrument, measured for 30 seconds, got the concentration. And we can see here they generated a theoretical concentration in the bag of 7.27 ppmV, measured it 6 times, 3 lots of replicates, and you can see here that we're getting RSDs for each set of 6 measurements of less than 0.5% RSD. Recoveries 99% plus recoveries for this. So clearly inherently precise instrument, inherently precise way of measuring formaldehyde. And when we do the statistics for all 18 measurements, again the RSD still sits below 0.5%. So as I said yesterday, an inherently precise instrument. Measure the sample time and time again, and if it's the same conditions, you get the same result every single time. Intermediate precision. So intermediate precision is essentially asking you to look at the system with somebody else using it, doing something else on a different day, hopefully with a different instrument. So conventionally you'll look at a different day, you'll use a different analyst to make the samples, you'll use fresh mobile phases if it was LC, which we don't do here. Fresh preps are the standard, different column, we don't have a column. Different instrument ideally. Now Joe would have loved to have done this on a different instrument, but he only had 1 in the lab. I'd have liked him to have 2 in the lab, but he only had 1 in the lab. So basically, he used a different analyst, different day, different sample prep, but the same instrument. But nonetheless, it's a different prep here. And again, prep the samples at the same 7.2 ppmV, run the samples again, and pretty much replicated what we saw the day before. Again, RSDs of less than 0.4%, recoveries of around about 99% plus. So precise. And when we stick these 2 measurements together to give like 36 measurements of formaldehyde in a Tedlar bag, incredible precision for this, and as I said yesterday, no internal standards. So I mean, it works really well. So obviously, as we said yesterday, this instrument is inherently precise, but now we need to check accuracy. So there is a ICH guidelines cover how to do accuracy. So it should be assessed on samples spiked with known amounts of impurity. So you spike in a concentration, and you hope to recover the concentration you put in and measure the right concentration. The issue with this method was they were looking for formaldehyde trapped inside pre-filled and pre-pressurized pouches of varying volume. The manufacturing process didn't give a fixed volume in these bags. So how do you spike in a known concentration that you can recover accurately if you don't know the volume that you're spiking into in the first place? So you can't actually do this spiked recovery exercise with a gas phase sample unless you can absolutely fix the volume of gas you're spiking into. And in this case, because they were manufactured products, you can't do it. So the approach taken here was not to spike in and recover at known concentrations, but repeat the precision test at 10 and 90%. So we're showing accuracy at the back ends of these, the expected range of the method. So here we see the data, so very similar to the precision data, 10% and 90% of the linear range of the method, and again we're seeing really good recoveries, so we're recovering at least 90% plus of the analyte being in the bag. I would say that looks quite accurate to me. Next step of the ICH validation process is robustness. So basically what robustness is talking about is, I'll quickly sort of go through this, is essentially what you're looking at is if the instrument has things and ways in which it varies over time that you can't control, does the method still give you accurate and precise results even though that variation is taking place? So what you've got to look at is you've got to look at your instrument and say what things vary that I can't control, and then rerun some test methods looking at that instrument in that modified state. So when we looked at the SIFT-MS, really the only thing that varied was the temperature of the inlet and the temperature of the flow tube, because they have a tolerance in which they move about a bit, so it's about plus or minus 7 degrees. And as this is a gas phase measurement where you're flowing gas through an inlet, the temperature changes on the inlet, your flow changes slightly. As it's a kinetic reaction takes place in the flow tube, if your temperature changes on the flow tube and your pressure changes, then your reaction rate changes. So this can lead to variability in your measurements. So what was done for the robustness was the flow tube and the inlet temperature were varied based on the tolerance that that instrument would sit in. So plus or minus 7 degrees on the flow tube, plus or minus 7 degrees on the inlet. And again, using a prepared bag standard from the gas generator, run the 7.2 ppm standard 6 times, look at the recoveries for that, and you can see again we're 90% plus recoveries for this. So even with this variation in temperature on the instrument that we would expect it to see over the measurement time of the method, we can see that you get very good recovery. So again, the method is actually robust. So we can expect good measurements even if there are small changes in the environmental conditions of the instrument. Next part of the validation exercise is looking at limits of detection, limits of quantification. Now, anyone that has used SIFT-MS knows that limits of detection, limits of quantification on SIFT-MS are somewhat of a movable feast. The longer you measure for, the lower you can push these limits. It's not set in the same way. Not really set, you know, as we would with a GC where you spike in a low level and see if you can see it, or you do 10 times the signal to noise. If we measure for longer, we can actually generate different LODs. So in this instance, what they chose to do was to run a system blank and essentially use the standard deviation of that measurement, 30-second measurement, and then look at 3 to 1 and 10 to 1, 10 standard deviations of that measured blank matrix sample. And came up with LODs and LOQs of 1 and 4 ppBV, which is so far away from the expected concentrations in this method that it's a nonsense, to be honest with you. But it needs to be done as part of the validation exercise. You need to be able to determine LODs. So the way you do it with SIFT-MS is you just have to define your process for LOD and LOQ and stick to your defined process, because it's going to be slightly more odd than it is for a standard chromatographic method. So that went through that LOD, crunched all the data, wrote it up, put it through their quality system, submitted it to the client had a look at it, and their summary of the whole method was it passed. So we now actually have a validated SIFT-MS method for a regulatory submission method for a pharmaceutical device, which is quite a bar to sort of cross really in terms of an analytical method. It's one of those things that requires a lot of work and a lot of confidence in your system that it's going to work to be able to do this. So I think for that type of method, for that analyte, this is a fantastic result really. And so I think it was the first time they'd seen a SIFT-MS method for these types of submissions. I'd be interested to know how the MHRA or the FDA would audit this, because it's going to be odd for them, but I suspect, you know, they'll have to get used to it, because there's going to be more of this stuff about, I'm sure. So just to summarise, or put Joe's summary in, that method validation on SIFT-MS is achievable following the ICH guidelines, but modifying them to your needs for your method. And there are some considerations such as what is a blank, how do you do your LODs, but we can, as long as we think about what we're doing and define what we're doing, we can actually get through a method validation. And so just to see, that's Joe at Intertech, so I'd like to thank Joe for that. It doesn't work there anymore, unfortunately, but nonetheless, it's all his data and it's all his work, and it's a very good piece of work. So now I'm going to move on to a piece of work I did in the lab. So this is a more complex method in the sense that it's got some sample prep up front, it's running calibrations in aqueous solutions and stuff. So it's a methanolic extraction of soils for BTEX. So BTEX compounds in soils is a particular environmental method that needs to be carried out, you know, around petrol stations and contamination around, you know, factories and stuff. And so there are many ways of doing this. And one method approach to do BTEX compounds in soil is a methanolic extraction approach. And this basically is how you do a methanolic extraction method for BTEX determination. So you take some soil, you add 6 mils of methanol to it in a headspace vial, and you sonicate it up to extract the BTEX compounds, and then you centrifuge it to pull the soil to the bottom, and you end up with your methanolic extraction at the top. You remove 250 microlitres of that methanol and you spike it into an aqueous or sodium chloride solution in a headspace vial, and then you incubate that new headspace vial, and then you measure the headspace. And so we've done this in the lab on GCMS, and so I thought I'll replicate it on SIFT-MS. So it's one of those examples of a more complicated method we can run on SIFT-MS using automation and using all the tools available to automation. So at the moment, we can actually automate that whole process there is fully automated. With the new MPS robotic and the ability to change syringes and use large volume syringes, we can actually do the methanolic addition as well as an automated process. So apart from filling up a vial with soil, the whole process is now automated. So we just put vials on the MPS, press go, get the whole prep and the measurement at the SIFT-MS. So it's an example of doing complicated method preps and running it through the SIFT-MS. So what we wanted to do was just basically say, if we had some soil, does this method stack up, or do we get completely random results? So I followed the sort of ICH type approach. So first things, of course, is to look at linearity. So I just made up some standards like you saw yesterday, BTEX and in water or in sodium chloride, run my linear curve, concentration range from about 10 ppb up to 2000 ppb in solution, and you can see we get some good linearity. But we did see the same thing we saw with that formaldehyde method. If you look again, this is another approach to plotting recoveries. We weren't recovering very good concentrations at the bottom end of this curve. In fact, we were under recovering this time. And again, this is to do with the top end curve, at top end points, pushing that bottom end away from zero. So we needed this sort of top range concentration. So what this is telling me is that actually my lower end concentration in solution is around about 15 ppb in solution. But that's fine, that's perfectly acceptable. So then a system precision. So in this instance, we basically produced some standards, some repeat standards at I think it was 5, 20, 200 and 2000 ppb in solution. Run the repeats at each concentration and then did the standard precision data for each of the analytes. And you can see here that at the low end concentrations where we're getting that slight under recovering because there are linearities, the RSDs are fairly poor, but they're okay. They're less, they're less than 20%, and I've done some methods where 20% was perfectly reasonable in the past. Pharmaceutical as well, so if pharma industries accepts 20%, I'll accept 20%. But up at the top end concentrations, the expected concentration range is here. Our RSDs are 5% or less, which again It's an inherently precise instrument. You know, we put the same sample in the same time, you get the same results coming out. If you recall from the methodology, once you've done your methanolic extraction, you take 250 microlitres out and you spike it into the water in the headspace. Now, if we want more sensitivity, we could spike more methanol in because there are going to be more BTECs going into your headspace file and you recover more. If you want less sensitivity because your top end concentration is too high, you could spike less into the water. But we need to understand, is that still linear? If I vary my methanol spike, is it going to be linear? So this plot here is basically running a stand, making a standard up in methanol, and then spiking various volumes of that methanol into the sodium chloride solution from 25 microlitres up to 500 microlitres, and then doing the analysis, checking the linearity. And you can see that, again, we get good linearity, 999 plus R squares for all of our analytes. So that does mean we have some scope now to vary our concentration range while still running the same range of the instrument, just change the spike volume of your methanolic extraction so we can make it deal with more concentrated samples, we can deal with less concentrated samples within our linear range that we've shown before. And just so the next part of the process is accuracy recovery. So what we do is we basically spike in known concentrations into our matrix and then try to recover it. In this instance, because the matrix is a solution of a fixed volume, I can do the spike experiment because I know the volume, so I know the spike, so I know the recovery. This is an example of recovering toluene at these various concentrations spiked into the solution. That's just the result you get from the SIFT-MS, the standard injection profile that you saw yesterday and probably saw in the lab yesterday as well. And so you can see good data. And then if we look at the results, if we spike in various concentrations and we run a linearity through it, we get back what we expect and we get back in a linear way. So again, the accuracy recovery, if we put it in the matrix, we get it out of the matrix, we get a good result coming back out of the matrix again. And just to show that there was a very good, you know, one-to-one relationship here, this is all of that data plotted, spike concentration versus calculated concentration from our linearity curve. And you can see the slope is essentially 1, so there is a one-to-one relationship, y equals x. So what we put in, we get out according to the linearity and the calibration we're running. This is, again, the recovery now of that accuracy and recovery exercise. So what are we recovering back from our spikes? Again, you can see at the low concentrations, we're slightly under recovering, but the concentration range we expect, our recoveries are somewhere between 90 and 110%. Fully accept a plus or minus 20% recovery. It's perfectly acceptable method. And then analytical precision. So again, let's make up some samples, but in analytical precision as opposed to system precision, what we're looking at is we're looking at our samples now. We're not looking at our standards. We're spiking into a sample or getting a real sample and running multiple replicates and seeing if we get the same result across multiple replicates. So this is a 200 ppb spiked soil sample now, and then run through the whole methodology, do the calculations of the calibration curve and see what we get for the precision. And you see we get RSDs of less than or about 1% or less for each of the analytes. So again, highly precise method. And just to show that we're recovering the right concentrations, these are the recoveries for that data. So again, we're recovering near to 100% what we expect to see. So again, precise, and also the methodology works for us. So the method works and the instrument allows you to make those measurements as well. So that sort of was a partial method validation on a completely different method to the gas phase one. But again, you can see from the data that it's entirely possible to validate SIFT-MS methods, even relatively complicated automated SIFT-MS methods, exactly as you would any other analytical method. So, you know, if you've got any concerns that SIFT-MS is a method to do analytical chemistry on, hopefully this tells you it is not, and we can actually prove it. We can do standardized methodology following ICH type guidelines and prove that these methods work and have a proven validation at the end of it. So I'll leave with that. Thank you very much.
Does anyone have any questions from
Someone must.
In general, I know it is necessary to go through with validation for a new method.
Mm-hmm.
However, when we want to use, say, ISO method, AOAC method, or EU method, do we have do the same?
Well, unless the method states that because if you look at ISO methods or many methods, it will say that it will give you the required technology to do it, and in many cases it will say or a validated alternative, and that's the critical part of it. Is if you're going to use an alternative method to what's stated in the ISO standard, you will have to do some form of validation. It may not have to be a complete validation exercise. It may be enough to show that if you could potentially run a side-by-side method, so run the ISO standard as defined, so it might be a GC-MS method, and that side-by-side run your SIFT-MS method and show that the results compare, and that might be enough to validate the use of a new methodology on an older ISO standard. But absolutely you're right that many standards are written with a particular analytical method in mind, and that's always a problem. But you do have scope to pull these things apart based on the line that says at the top, or a validated alternative. And then it's up to you to decide what your validation process is. What do you need to do to prove to your auditors or whoever is looking at the methods and the answers you're producing that this method is giving the right result for this new technology, basically.
I've always got questions.
You always have, yes.
Great talk, really enjoyed it again, and that's really promising because that's actually not that bad, I don't think.
No.
It's definitely doable. Anyway, what I was going to say was one possible stumbling block is, what if you do have a conflict in the library?
Well, I don't know. I mean, basically, if you do have a conflict in the library, then I suppose you're going to have to do some more work on your method in the first instance, understanding your matrix, and then proving to yourself and those people who need to be proved to that the likelihood of this conflict appearing in your matrix is so low as to not be a problem. But I mean, it's like anything. You can do your specificity on a GC method and look for co-elutters and such like, and they're not there. But there's always a possibility that you get a sample coming through that is slightly odd, and you have a co-elution that you don't know about. And then what will happen, of course, is hopefully that your method is designed in such a way to capture that deviation. It will throw off the result in some manner that you'll say, well, there's something odd going on here, and then you'll have to do some sort of deviation investigation as to why this is occurring. I think one of the things to say about the specificity is that what we have going for us with SIFT-MS is that because we can use the multiple reagent ions, it actually will throw up matrix interferences relatively easily, because you will see if you use 2 reagent ions to measure, or 2 product ions to measure the same concentration, if one's got an interference, you'd hope that you wouldn't get interference in both in the same way. You would find that deviation in measured concentration, and then it's up to you to decide what's going on.
So do you think you could make that case in the validation to say, we have got these conflicts on these ions with this precursor, but if you take the whole 3 and maybe your secondaries,
I think you can, yeah. I mean,
you have to have another stage where you said, okay, we'll do an experiment where we put in some mixed samples.
Yeah, possibly. I mean, I think you can, my feeling is, having done this type of stuff for years and years and years, is you can pretty much do anything you like, as long as you say what you do what you say, and can justify it to the person that's asking you the questions. You can get into some quite robust conversations with the person asking you the questions, but if you have confidence in what you do, then that's fine. So, yeah. Make sure your method, that all comes in the method development part of it. You've done enough development that you have your arguments set already for the conversation you're going to have with your auditor. And you tell them to go away, you're wrong. That's what you do. They don't like it when you do that, but it has to be done.
I've got one quick thing. So the gas standard generator, had that previously undergone some sort of validation?
Yeah, that had been done by, yes. Of course, there's the endless line of validation that runs back. So can you validate your labs the right size, and you got the right temperature, and is your thermometer, it goes back and back and back. But it is about, again, it's part of your method development and what your requirements are.
Also, it's quite standard in GC-MS to put a 1/X fit when you've got your high level. Would that...
I don't know.
To needed a large dynamic range,
I don't know. It'd be worth looking at it. Be worth looking at it. I'll get my Excel spreadsheet out and do it in a minute.
Yeah.
Yeah, yeah.
I did have another question actually. So what if you can't easily generate a standard? I mean, that's potentially possible, isn't it?
It is potentially possible, and again, that will come back to how you develop your method and how you build your arguments in to ensure the robustness of your method. I think, yeah, if you can't get a standard and you're relying on the library and the K values and stuff, what's your...
That's really worst case scenario, but
Yeah.
I suppose you could, if you've got another method that can measure whatever it is, then you can...
Yeah, I mean, you've got...
There's two methods that measure that,
Yeah.
therefore we're comparing those, but some cases even that's difficult.
It is. I mean, I like to think that sometimes it is difficult, and that's a good thing, because if it was easy, anybody would be doing this. Yeah. I like it difficult because it means only some of us can do it. Keeps us in a job, you know, pretty much. Anyway, yeah.
Great.
Yeah.
Thank you, Mark.
Let me pull the next one up.